So this week we're going to take a look at how we use bioinformatics to analyze protein sequences. But before we do that, we're going to do a little bit of review so we have perspective. Remember the function of DNA. DNA is a molecule that's used to store and transmit information in the form of genes. And hopefully you remember the central dogma of biology that tells us that DNA codes for RNA, which then codes for protein, where we go from DNA nucleotides to mRNA nucleotides to amino acids. So we change languages. So we go from DNA to RNA by transcription, and then RNA to protein by translation. Remember, this process then is called expressing a gene. So how often something is transcribed and translated is gene expression. Here we're looking at a strand of complementary DNA. I want to just remember, or make sure we remember, that DNA is anti-parallel. So remember from Honors Bio, anti-parallel is that it runs from 5' to 3' on one strand and 3' to 5' on the other. And it's complementary, meaning that cytosine always pairs with guanine and adenine always pairs with thymine. So this is just a template strand of DNA, where one side is the side that's read by DNA polymerase and is then transcribed and translated into a protein, and the other side is simply the template strand. So as we look at this, we have our gene coding strand on top, or our template or non-coding strand on the bottom. Now, we don't always know which strand is which when we look. Actually, frequently we don't know. So instead of saying don't always, we don't know which strand is which is the coding or non-coding strand. Here we've just labeled them black and red so you can see the difference between them. But I want to just make sure we remember that transcription translation process from Honors Bio. So remember, we take our gene coding strand and we are going to then, from this strand, we're going to transcribe it into mRNA, which is then translated from the 5' to 3' direction into an amino acid, a complementary amino acid, or complementary protein rather. Each codon, remember each group of 3 mRNA nucleotides is one codon, and that codes for one amino acid. Here you'll notice in the mRNA, the start codon, AUG, has been circled. Remember, in the process of translation, we look up each codon and see which amino acid it codes for, or it stands for or it represents. Here, each of those amino acids has been abbreviated into one letter. So for example, if you look at AUG, which you know is the start codon, so right here, AUG, which you know is the start codon, also codes for methionine, so the amino acid is represented by this single letter. We can also use this particular chart as we take a look at our amino acids. This chart might be a little bit more confusing, but it's the same idea, where if we were trying to find, say AUG, our start codon, start in the middle with A for the first letter of our codon, then U for the second letter of our codon, and then G for the third letter of our codon, and that codes for methionine, which we abbreviated on the previous slide as M. I'm going a little bit quickly through this because I'm hoping that you remember doing it at the end of the year in Honors Biology, where we took a strand of DNA and then had to find the complementary mRNA bases, and then from the complementary mRNA bases we had to find the corresponding amino acids. You'll notice that each of these amino acids has been color-coded for the particular chemistry or characteristic of that R group. And here at the bottom of our slide, our amino acid structure hasn't been scrambled like it was in that last slide, so I apologize for that scramble. So let's just pick a codon. If my mRNA were to read C, C, U, it would code for the amino acid proline. The amino acid proline has a nonpolar or hydrophobic R group. The reason that this is important is because the chemistry of the R group influences the shape of the protein that gets made. Remember, polar things are going to avoid nonpolar things, and positive things are going to be attracted to negative things. So this being true, what that means is as our protein gets its three-dimensional structure, it's going to change based on what amino acids there are, because that influences how they fold, what chemicals or what R groups want to be near each other or want to be as far away from each other as they possibly can be. And as far as proteins are concerned, for protein, structure dictates function. The way that a protein is shaped is going to very strongly influence how that protein functions. So if you change its shape, you change its ability to function. One thing I want to take a look at that we didn't have the opportunity to in honors biology is the difference between the amino acids. Remember, there are a lot of different amino acids, and what makes them different is their side chain or their R group or their variable group. So this kind of looks a little bit scrambled, for which I apologize. You might recognize our central carbon, our carboxyl group that we can find on every amino acid. Oh, that one doesn't go there. Let me erase that. Oh, won't let me. There we go. There we go. Our amino group, and then our R group, or our variable group, up there at the top. The difference between each amino acid and what gives them a different name is what attaches at the R group. What attaches at the R group can be a variety of different things. Here we've highlighted the differences in this table by showing you that sometimes R groups are hydrophobic or hate water. Those are our nonpolar R groups. Sometimes they're hydrophilic or polar. Sometimes they're acidic, which means they have a slightly negative charge, and sometimes they're basic, which means they have a positive charge. So if we go back to that table on the previous slide, so what we have to do then, as we look at a sequence of DNA, is determine where in that sequence of DNA a gene is going to start. We call this finding a reading frame. So we're going to pretend that this first set of letters right here is a gene sequence. So it's something that codes for a protein. If we know that we read 3 letters at a time, the first reading frame is going to start at the first letter. We could read this as the cat ate the rat. Now, of course, our brain wants to read it that way because we know these are English words. So automatically this makes sense to us. But remember, in DNA, it's not always obviously the first letter. Sometimes the reading frame starts with the second letter. So if we were to do that with this example, the cat ate the rat, we would skip the first letter and say, well, that's not part of the gene in question. That's just the letter that happens to occur right before it in the genome. And instead, we have HEC, ATA, TET, HER, and then the last 2 letters wouldn't be included because they're not a full codon. So that's the second possible reading frame. There's another possible reading frame of the same sequence here that starts at the third letter. So we skip the first 2, say, oh, those are just 2 nucleotides that happen to occur. It could be ECA, TAT, ETH, ERA, and then a T at the end. So we have 3 potential reading frames on our first sequence, on our template sequence. We have 3 other reading frames that occur on the frame or on the sequence that is complementary. So remember, we said we didn't know whether we read the black or the red of our initial example sequence. if we happened to have read the red instead of the black, we would have 3 more reading frames on that bottom complementary for a total of 6 possible reading frames per gene. So every gene has 6 possible reading frames. The correct reading frame is what we call the open reading frame. It's the one that actually codes for protein. So because we recognize this sentence, we know that the open reading frame here, which sometimes we call the ORF, is the cat ate the rat. So I want to take a look at this and see if I can't help you guys see where our open reading frame or where our reading frames are here, where our potential reading frames are. I'll change my color to yellow. Here's my first potential reading frame right here. We start with the first letter. There's one codon. There's another codon. Third, fourth, and fifth. So that's one potential reading frame. The next potential reading frame would occur one nucleotide in. So there's one reading frame, or there's one codon, there's a second codon, a third codon, a fourth codon, and we wouldn't read those last 2 letters. Our third potential reading frame would be starting at the third nucleotide of our sequence. There's one, would start here, 1, 2, 3, and then 4. So there's the 3 potential reading frames on the top strand, and we would have 3 more potential reading frames on the bottom strand, remembering that DNA polymerase only reads from 5' to 3'. So our first reading frame on the bottom would be here. There's 1, 2, 3, 4, 5 potential nucleotides in that reading frame. Then there's 1, 2, 3, 4 potential codons in that reading frame. And then our third reading frame on the bottom starts at the third nucleotide in. So there's 1, 2, 3, 4 potential codons for that reading frame, for a total of 6 possible reading frames. It's possible to have so many reading frames or so many potential ways to read DNA because remember, our start codon is AUG, but AUG doesn't always indicate the beginning of the gene. Sometimes AUG is in there because it's coding for the amino acid methionine. So we can't interpret every AUG that we find as the beginning of a new gene. We would have too many potential genes in the genome. I know that it's very difficult to think about because I didn't introduce the idea of different potential reading frames to you guys during Honors Bio. But it's important to see that it is possible to read the same DNA sequence in multiple different ways. So what we do in bioinformatics is we take those whole genome databases that we've been working with and we run it through some software. Nobody wants to have to take an entire genome of 6 billion base pairs and determine all 6 of the potential reading frames and then translate all of those. So some software developers or actually it's not even software developers, it's biologists who are also computer scientists, like we're asking you guys to be, have developed different search algorithms to create what we call an ORF finder. And what that does is locates on every strand of DNA the 6 potential reading frames and finds the one that is highest probability of being the open or the correct reading frame. The open reading frame is often the one that produces the longest potential gene, or the longest potential, the longest potential gene, the longest potential strand of amino acids. So that's what that ORF finder does. In your lesson this week, you guys are going to use some data that you've previously analyzed to determine the open reading frames for your unknown organism, and then you're going to compare, using those open reading frames, the genes that are produced to determine if there's a change in the amino acid sequence and whether or not that change in the amino acid sequence changes the shape of the protein. So you can find that particular lesson attached on the Data and Design website. I'll link that for you on the Google site. I'll link it on the Google Plus site so you can follow those instructions. Good luck!